My name is Joshua Mugabi. I welcome you to Topic 2, Nutrition in Plants. Our learning outcome A, evaluate the relationship between the structure of chloroplast and photosynthesis in C3 and C4 plants. Details of biochemistry are not required. Our learning activity, we are going to be using role play. Learners in their groups demonstrate the light-dependent and independent stages of photosynthesis in C3 and C4 plants. We are going to look at photosynthesis in C3 and C4 plants. Item 1. Our emphasis now is going to be on the field observation of crop performance. A farmer observes that maize, a C4 plant, grows better than beans, a C3 plant, during hot, sunny conditions. Students are asked to explain this using knowledge of light-dependent and light-independent stages of photosynthesis. Tasks. Name the two main stages of photosynthesis. State where each stage occurs in the chloroplast. Explain why C4 plants perform better than C3 plants under high temperatures and light intensity. Name two main stages of photosynthesis. There is the light-dependent stage and light-independent stage, which we call the Calvin cycle. State where each stage occurs in the chloroplast. Light-dependent stage occurs in the thylakoids membranes, which we refer to as the grana, and light-independent stage occurs in the stroma. Explain why C4 plants perform better than C3 plants under high temperature and light intensity. C4 plants reduce photorespiration by concentrating carbon dioxide. They use PEP carboxylase, which has a higher affinity for carbon dioxide. This increases the efficiency of the Calvin cycle under high temperatures. We are still in photosynthesis in C3 and C4 plants. Item 2. Now we are going to emphasize on investigating the light-dependent stage. Students illuminate chloroplasts in the laboratory and observe production of ATP and NADPH. Task. State the inputs and outputs of the light-dependent stage. Explain the role of light energy. State the importance of this stage to the light-independent stage. State the inputs and outputs of the light-dependent stage. Inputs: there is light energy, water, ADP and inorganic phosphate, and NADP+. Outputs: there is ATP, NADPH, and oxygen. Explain the role of light energy. Light energy excites electrons in chlorophyll. They lead to photolysis of water. It drives ATP synthesis and reduction of reduced oxidized NADP. State the importance of this stage to the light-independent stage. ATP provides energy. NADPH provides hydrogen for carbon fixation. We are still under photosynthesis in C3 and C4 plants, item 3. We are going to emphasize on light-independent stage in C3 plants. Students study carbon fixation in C3 plants, such as rice and wheat. Tasks: name the enzyme involved in carbon dioxide fixation in C3 plants. State the first stable product formed. Explain why photorespiration occurs in C3 plants. Name the enzyme involved in carbon fixation in C3 plants. The enzyme is Rubisco, ribose 1,5-biphosphate carboxylase / oxygenase. State the first stable product formed. 3-phosphoglycerate, 3-PGA. Explain why photorespiration occurs in C3 plants. Rubisco also binds oxygen. At higher temperature or low carbon dioxide, oxygen competes with carbon dioxide. This leads to photorespiration, reducing efficiency. We are still under photosynthesis in C3 and C4 plants. We are on item 4. We are going to emphasize now on light-independent stage in C4 plants. Students examine examine leaf cross section of maize and observe crans anatomy. Task: name the two types of cells involved in C4 photosynthesis. State the enzyme responsible for initial carbon dioxide fixation. Explain how the C4 pathway reduces photorespiration. Name the two types of cells involved in C4 photosynthesis. The mesophyll cells and bundle sheath cells. State the enzyme responsible for the initial carbon dioxide fixation. PEP carboxylase. Explain how the C4 pathway reduces photorespiration. Carbon dioxide is first fixed into a 4-carbon compound in mesophyll cells. Carbon dioxide is released in the bundle sheath cells around the lubicle. High carbon dioxide concentration prevents oxygen binding. We are still in photosynthesis in 3 and C4 plants. Item 5. Emphasizing on comparative analysis of C3 and C4 pathways. Students prepare a summary table comparing photosynthesis in C3 and C4 plants. Task: compare C3 and C4 plants in terms of first product of carbon dioxide fixation, enzyme used, photorespiration, leaf anatomy. State one advantage and one disadvantage of the C4 pathway. Compare C3 and C4 plants in terms of first product of carbon dioxide fixation, enzyme used, photorespiration, and leaf anatomy. In our table, we have the future C3 and C4 plants. The first product in C3 is 3-phosphoglycolic acid, a 3-carbon compound. In C4 plants, the first product is oxaloacetate, a 4-carbon compound. The enzyme in C3 is lubicle. The enzyme in C4 is PEP carboxylase. Photorespiration is high in C3 plants and very low in C4 plants. Leaf anatomy: no crans anatomy in C3 plants. Crans anatomy present in C4 plants. State one advantage and one disadvantage of the C4 pathway. Advantage of C4: high photosynthetic efficiency in hot and dry conditions. Disadvantage of C4: requires more ATP. Marking emphasis: correctly describe both stages of photosynthesis. State correct locations and products. Distinguish clearly between C3 and C4 pathways. Use correct enzymes and products. Apply knowledge to environmental conditions. Conclusion: go to the next learning activity. Thank you for participating in Topic 2, Nutrition in Plants. Learning Outcome A: evaluate the relationship between the structure of chloroplast and photosynthesis in C3 and C4 plants. We said details of biochemistry are not required. Our learning activity has been Part B. Using role play, learners in their groups demonstrate the light-dependent and independent stages of photosynthesis in C3 and C4 plants. If this has been very useful to you, please contact me at Mugabi Biology on YouTube.